Okay, we're live here. So what we want to do today is finish up chapter 2 in terms of the nucleotides and the nucleic acids. We've covered all the rest of that chemistry in chapter 2, and this is the end of chapter 2. And I'll close that out in a little bit, and then we'll get into chapter 3. So as you look on page 59 of your textbook, page 59 of your textbook, halfway down the first column, you see nucleotides and nucleic acids. Make sure you do the, don't worry about flashback on this one. I was going to say to look at it, but don't worry about it. We're not going to cover all that. So you see the bold print terms, nucleotides and nucleic acids. When you think of nucleic acids, you think of two things. Think of DNA and RNA, deoxyribonucleic acid and ribonucleic acid. Both are needed for us to stay alive. The deoxyribonucleic acid contains our genetic makeup. That's why we look the way we look. Some of the skills we have anatomically or whatever, all due to DNA. Or deoxyribonucleic acid, and that's in the nucleus. It always stays in the nucleus. And then if you come down, you see in the italicized form ribonucleic acid, which is another kind of nucleic acid, but it plays a role in protein synthesis. DNA is a code for thousands and thousands of proteins that have very specific functions. We have to make those, and ribonucleic acid plays a role in that. We'll get into that a little bit later in chapter 3. It's an amazing series of events that we take for granted. But anyway, there's also a third type of nucleic acid. It's really not nucleic acid. It's called a nucleotide. And as you come down into that paragraph, you'll see the abbreviations in a parenthesis. It says ATP. We'll talk about that in just a few minutes. So looking on the second column, page 59, you see nucleotide structure. Each nucleotide has 3 components. And if you would, look down in the middle of that column on page 59, second column, page 59, you see a nice little drawing there, illustration. It says the structure of a nucleotide. 3 parts. So you can see the little hexagon, and that represents a nitrogenous base, which we'll talk about in just a second. And then you see the pentagon, and that is ribose, the sugar. In this case, if it's in DNA, it's deoxyribose. But in this case, we'll say it's ribose. And then you see a phosphate group, sort of like we had back with the phospholipids. Got to have some phosphorus in our diet. Now, as you look below that illustration, you see there are 2 boxes. One box says purines, second box says pyrimidines. And in the purine category, you see these 2 nitrogenous bases. You can see there's a pentagon, and you can see there's a hexagon attached. They're not identical. In that structure, they are identical in having a hexagon and a pentagon. But if you look carefully, you'll see that there are different elements at different parts on those ring structures. And you want to know those, adenine and guanine. I'm not going to ask you to draw them. Don't worry about that, okay? That'd be into a course in biochemistry, which you may get as a senior if you go on to school or you get into grad school. So anyway, that first group, purines, contains 2 nitrogenous bases, adenine and guanine. And you come down to the bottom, you see pyrimidines. More letters, right? More letters. And you see there are 3 pyrimidines. And they don't have that pentagon attached to the hexagon. They simply have a hexagonal shape, six-sided shape. And again, if you look around, you'll find out that the elements associated with the corners are different. And that makes a big difference in terms of whether it's cytosine or thymine or uracil. So you've got these two categories of nitrogenous bases. Now, how are you going to keep all that stuff straight? Well, this has helped a lot of kids. Purines, that word has fewer letters, doesn't it? So when you think about purines, think of only two nitrogenous bases. And you look at pyrimidines, and you see there are three. Pyrimidines has more letters, therefore you have more nitrogenous bases. Maybe that'll help you a little bit, but it can go even further. You've got adenine and guanine in the purines. Think about the abbreviation for the state of Georgia, GA. Guanine and adenine. Maybe that'll help you keep it straight. Down at the bottom, you got three pyrimidines, cytosine, thymine, and uracil, but spell the word cut, C-U-T. And maybe that'll help you remember the cytosine and uracil and thymine. So the basic structure of a nucleotide, as you see right above there, it's always going to have a phosphate group. It's always going to have a 5-carbon sugar. The difference can be, if it's an RNA nucleotide, the difference is going to be the type of nitrogenous base that's attached. If it's DNA, going to have DNA as the 5-carbon sugar, and then you'll have these nitrogenous bases. Now, let's go over to page 60. And on page 60, you see there's the first column, adenosine triphosphate. That is another nucleotide that is used to carry energy for our metabolic needs. If you look below at the figure, you see the adenine, the 5-carbon pentagon, and then the 6-carbon hexagon. And then you see ribose below, that's a 5-carbon sugar. And then you see the combination of the two right below says that's adenosine. Then you come down, when you add a phosphate group, you get adenosine monophosphate, mono meaning one, adenosine diphosphate, that's a second phosphate group that's added. And if you add a third one, you get adenosine triphosphate. That is our source of energy for making chemical reactions occur in our body. So tonight when you're asleep and you had a supper and your stomach is working and breaking those things down so you can absorb the food, and you're going to turn a lot of it into adenosine triphosphate. That's our currency for energy. If we don't have ATP, we're dead in the water. As you look at the top of the second column, you see it says a molecule of ATP may last only 60 seconds. We're using it all 24/7. Even when we go to sleep, we turn and roll around like that, and we have to ventilate. We ventilate because of the diaphragm. Those of you who've had 110, you know that, and maybe some of the others of you do that too. But the diaphragm has to contract and increase the chest cavity, make it longer, and it draws air in, and then we relax. We've got a little system up here in our brain. We'll talk about it later when we get toward the end of the semester. And it turns on, turns off, turns on, turns off. You don't have to think about it. All you got to do is just go to sleep. You and I go to sleep by faith every night that that's going to work. And it does. So As you read that last paragraph, you'll come down about 3 or 4 lines from the bottom, and you'll see it says oxygen is required for most ATP synthesis. So we've got to have oxygen in order to make ATP. And you see it says the need for ATP is the main reason we breathe oxygen. We stop breathing in several minutes, we're going to die. So we have to make it all the time. And of course, you breathe harder when you're out there exercising. Now, let's go away from ATP. We'll mention it, well, you'll probably get it in the second half, Bio 211, when you get into the GI tract, into the Krebs cycle and all that wild stuff that goes on that we don't think anything about, but it's amazingly complex. Anyway, second column, the nucleic acids, DNA and RNA. Now remember, I said DNA is the molecule that is the blueprint for our genetic makeup. And you've got all these different genes, and they code for proteins. And remember how we said proteins are specific in what they do. Lactase, which we talked about back in the beginning of chapter 2, is an enzyme that breaks down lactose into galactose and glucose so that we can use it for energy. Lactase will not do anything else. It's made specifically for that purpose of breaking down lactose. The blueprint for lactase is in the genes. We've got to get a protein made. And so it takes not only deoxyribonucleic acid, which is the code for 100,000 different proteins. There's also ribonucleic acid, and ribonucleic acid is involved in converting the code into a useful, specific protein. So your DNA, as you see in the second paragraph, says is a polymer. It's linked. One nucleotide after another, sometimes billions of nucleotides long, but we get it into this tiny little nucleus. Great use of space, isn't it? Got all that knowledge. People say that there's enough information in our nucleus to fill up something like the Encyclopedia Britannica and more. Anyway, notice it says that the nucleotides in a nucleic acid are linked by dehydration synthesis. There we are, already going back to the first part of chapter 2 on chemistry. So what's another term that describes dehydration synthesis? How about anabolism? That's where you're building a bigger molecule. See, you got 2 terms that'll just tell you about the same thing. Now let's look over on page 61, and you've got these illustrations. As you look on the left, up at the top, you see what looks like a couple of purple ribbons that are entwined with each other. I call them the ladder, the rails of the ladder. If you think of a ladder, it's got 2 rails and it's got steps going up. So some people describe it as a ladder, and I've done that before. Some others may have another way of doing it. But anyway, DNA is a double helix. Look at the bottom of the page, page 61, and you see the bold print term in the first column, second paragraph, double helix. Now I want you to look at the twisting purple ribbons, if you want to use that word, or rails, and then look at the steps going across. You see T attaches to A, C attaches to G. Those are the steps. Now they untwist that a little bit, and here in the box below is a picture of that item above, of the DNA, section of the DNA above. Looking on the left part of the rectangle, you see a phosphate in that purple area. hooked to a pentose. In this case, the pentose is going to be deoxyribose. And then you come on down a little bit more, you see another phosphate, and then you see the pentose deoxyribose. So the rails of DNA, or you can use the sides of the helix, however you want to describe it, it's okay, are connected, or deoxyribose connected to phosphate groups. That's what the rails are made out of. The steps, which you see going across to the other rail, those steps are made of the purines and pyrimidines. Notice the G, in that first step, working our way down, the G attaches to the C. Guanine attaches to cytosine. It doesn't attach to anything else. And notice the type of bonds that hold those two nitrogenous bases together. Hydrogen bonds. You ever heard of those before? Me too. Important. So now you start thinking, oh, that's a polar covalent type thing there, because you know where those hydrogens are not, the electrons of the hydrogens are not equally spread out in the oxygen. They're not equally donated or shared, rather. They want to donate it, that's an ionic bond. But you come on down to the next one and you see adenine and thymine. Adenine always combines with thymine. In DNA. So you can see what the rails are made out of. The deoxyribose and the phosphate. The steps across are made of nitrogenous bases. So the G, which is a purine, attaches to the cytosine, which is a pyrimidine. Down below that, the adenine is a purine, which attaches to thymine. It always does that. Now you look to the right, up at the top, and you see single-stranded RNA. DNA in humans and dogs and plants, always double-stranded, but RNA is single-stranded. And if you look on RNA, you'll see coming down from the top, you'll see the G. I think it's a U, isn't it? Yeah, it's a U. That's uracil. Uracil is only involved in making RNA. So you got uracil, cytosine, adenine, guanine, uracil. Notice there is no thymine in there. Thymine is always found with DNA. So you want to keep that in your mind. And you come over to page 62. And what I want you to do is read in the first column, come down to the second paragraph. It says, when the DNA, we find the genetic code. Read that, and over on the next paragraph. We're not going to get into protein synthesis at this moment. We will get to it when we get to the end of the chapter 3. But I want you to notice as you come down the second column, and you see it says in the second paragraph, RNA plays a crucial role in protein synthesis. And so they give you some pictures of how to remember that. But you come down almost to the end and you see the terms in italics, transcription and translation. That's the two processes that are involved in making a protein. I'm looking forward to, we get to that point. I'm always amazed at how this goes on. And it's been working, in my case, for 73 years. Maybe in your case for 18 years or 22, 23 years or something like that. Transcription. Look up the word transcribe. Look it up and get a definition for it. Then translation. Look it up, get a definition for it. Those are the two processes that are involved in protein synthesis. Now, in closing this lecture out, I want you to make this note. I think some of it is in the handout, maybe in chapter 2. I know it's in chapter 2. And I want you to look over these questions. See if you can answer them. If you can't, look them up in the appendix. Okay? Can you imagine some of the questions on the test next Thursday? Not this Thursday. Listen. Not this Thursday. The following Thursday. I want you to check off number 1 under level 1. This is in the second column on page 66. It says assess what you've learned. So you see level 1. Check off number 1. Check off number 2. Number 3. Number 4. Number 6. Number 7. Come over on page 67 and check off 11, 12, 13, 14, 15, 16, second column, 17, 18, 19, 20. Level 2. Check off number 1 and number 2 under level 2 of check your understanding. Okay, well, I think we did that in about 22 minutes. So make sure you read the Word document at the end, and these are all there at the end of the Word document. And what I'm going to do is close out now, and probably tomorrow I'll start chapter 3. Send you a video on that also. And I'll send along with you a Word document, along with that video, a Word document. Okay? All right, I'm going to close out and see you tomorrow sometime. Bye.